the Journal of Child Psychology and Psychiatry, celebrating 60 years of empirical research, clinical studies, and systematic reviews. For details of our 60th anniversary celebrations, visit www.acamh.org/JCPP60. Follow us on Twitter with the handle @thejcpp and use the hashtag JCPP60.
Years ago, we didn't have any genes that were associated with complex behaviors. And we had the power to detect some large effects, but we were beginning to think that maybe the biggest effects were a lot smaller than anyone thought. And I did think back then that we could at least put them together like items on a scale and predict even if there were lots of genes of small effect. 10 years ago, at the 50th party for the journal, the first genome-wide association studies were being done, and for the first time we really were getting replicable associations, but it proved that the biggest effects are incredibly smaller than anyone ever thought, far less than 1% of the variance, and that meant that to account for heritability you'd need hundreds, and we now think thousands, of inherited DNA differences to account for heritability. But even so, you could put them together in sets of genes, and you could use them to ask important research questions about development, multivariate issues, and interplay between genes and environment. So you notice that first paper was called "When Are We Going to Be There?" So when are we going to be there? And the answer is now. How are we going to get there? Large genome-wide association studies. And where is there? And this is the point of my talk. I think it's polygenic scores. So I won't have time in my 10 minutes to give you all the background here, but you all know you can get DNA from any tissue. Every cell from the first cell with which we began life and from which we now have trillions of cells all have the same DNA. So you can get DNA from any cell, mostly from blood or from urine, but often in these kits from saliva. Then you genotype DNA differences throughout the genome. SNPs is one type. It's a single nucleotide polymorphism. And you genotype hundreds of thousands of them spread out throughout the genome, and you correlate each one with some trait. It could be reading ability or schizophrenia, autism. And you have to correct for 1 million multiple tests. But even so, those genome-wide association studies, once they got large enough to detect these small effects, are detecting a lot of significant hits. And then, what are you going to do with effects that are so small? It's like items on a test. You don't have a scale with just one item. You put a lot of items together. And together, they can often be importantly predictive. And that's what we mean by a genome-wide polygenic score. They're generically called polygenic scores. I call them genome-wide to distinguish them from just taking a bunch of, say, candidate genes and putting them together. It's an atheoretical approach looking across the genome, genome-wide. And the second reason is I can never resist a good acronym, GPS. Okay, so we have hundreds of these polygenic scores now. Most of the work has gone on with major psychoses. Schizophrenia, we can predict with DNA alone 7% of the liability. But the surprise in the last 2 years has been educational achievement. We can now predict 15% of the variance in GCSE scores with DNA alone. Childhood behavioral problems haven't had the funding that the big boys have had, but even with here's what you're talking about, 20,000 cases, they're still explaining almost as much variance. ADHD, perhaps 6% of the variance. And it is the variance. You can use a continuous measure. These aren't just diagnostic specific. In fact, they're contrary to that. They're saying there are only dimensions. So if the future is here, what's the future? And I think it's better polygenic scores. And how are we going to get there? Well, most of the life sciences deal with this. It's called the Missing Heritability Gap. And what people mostly mean by that is the gap between how much variance we can predict with known GPS, polygenic scores, say like in the case of reading, we're talking about 10% of the variance, versus how big the heritability is, which is typically 50%. That's the gap people talk about. But importantly, there's two types of missing heritability. the GPS is limited to the SNPs that are on the chips that we use to genotype. Those SNPs are common SNPs. Most DNA variation is rare. By common, we mean 1% of people have it. So that's missing GPS heritability, and the way to beat that is to have, if we have larger samples, we can get up to 25% heritability, but the current chips, we can't get beyond that. That's the ceiling. So how are we going to get to twin heritability? And most people believe that we're going to have other DNA differences. And the way it's going to go is whole genome sequencing. There you've got all 3 billion base pairs of DNA, not just a sample of a few 100,000 DNA differences. Okay, so that's what everybody's doing in the life sciences. But for us, I think what's far more interesting is to get better polygenic scores that are more specific. So the winner of all these polygenic scores is the absolute worst, coarsest variable you can imagine, years of education. But because it's included in every GWAS study, you get samples of over 1 million, and as a result, you get a polygenic score that predicts everything better than anything else. It predicts mental health better than anything else, because what does it take to get through university? It doesn't just take intelligence or achievement, it takes stability, persistence, and mental health. But wouldn't it be great if we were able to look at more specific cognitive abilities, STEM subjects? We could look at these things independent of G, which interests me a lot. We could also do that for behavior problems. I mean, as Timmy was pointing out, a bit subtly, I think, but the point of it is that we really need to take this P idea seriously. But we can look for specific problems independent of P. And we can do age-specific, environment-specific, treatment-specific polygenic scores. But to do that, we're going to need these huge GWAS studies. So how are you going to do that? Well, the way things have been done so far, like with educational attainment, is to just put lots of studies together and meta-analyze them. But then you have diversity of samples and measures, and how are you going to get something specific, like reading-specific problems? One way is through the national biobanks, like the UK Biobank. Genomics England is now going to try and sequence 1 million people. US has started a project with 1 million. But you still got to get people tested in those samples, and it's going to be difficult. Direct-to-consumer testing, over 15 million people have paid to have their genotyping done. And 80% of them agree to participate in research, and a lot of that is psychological research. So that's another angle. But here's my big reveal. How are we going to do it? I think we're going to have universal genotyping on the NHS. And we already do it for neonates. We do it badly. We only genotype a few genes. We ought to be doing SNP chips, no question about it. But more than that, I think we ought to make genotyping available for everyone. Finland has begun to do this. It'll be done for health. You can predict cardiovascular disease better with DNA than anything else. 8% of the UK population is walking around with a 3-fold greater than average risk for having a heart attack. You can prevent this. You can prevent a lot of disorders, and DNA is the best predictor in town. It'll be done for medical disorders, but once you do the DNA, the genotyping, you can get any of these hundreds of polygenic scores. So alcoholism, you can't become alcoholic if you don't drink a lot of alcohol. Obesity, well, we all know you got to prevent diseases, and this is the way to do it. So I was going to say, with universal genotyping, the NHS, I'm surprised to see people go, oh my God. But I thought it was the big reveal. Then did you see last week's Times front page? Gene tests for sale on the NHS, being Tories, of course they'll charge for it. But actually it's not we can discuss this later. There's a lot of things to discuss later. But the Twitter sphere went into fibrillation with this. So I decided I'd write something. I wrote it for The Spectator on the huge advantages for the NHS if this could be done. And so I hope we can discuss these sorts of issues later. You got a phenotype too, though, if you want to do a GWAS. But again, the NHS has these electronic health records. I know there's problems, but there's a lot of data there that other countries wouldn't have. And people may not know that the National People Database has data on 26 million kids in the UK, a lot of data. And you can do web-based collection, and get a lot of data on specific things, not just coarse variables, and data mining without even contacting people. You can get an awful lot of information. So I think that universal genotyping is going to transform how we do research, because any subject you study will already have their genotyping done. And eventually then that will also transform the clinic. So you think about the advances in the last 10 years, the mind boggles at what we're going to be talking about 10 years from now. Thank you. Let me introduce the next speaker. It's a great example of JCPP. Brad Peterson, we were just having coffee, and I was telling him about the study I was going to design. He listens 5 minutes, and he says, actually, if you did it this way, it would be a lot better, completely different from what I was going to do. And absolutely right. So thank you, Brad.
To be part of the advancement of child and adolescent mental health, visit www.acamh.org.